Hello dear students. Today we will study about a very important type of biomolecules which are actually the tools in the field of biology. They are the biocatalysts who regulate every reaction in the living organisms. Yes, we are talking about the biocatalyst enzymes. So we will be studying these particular topics in enzymes, which are, we will study some little bit about the discovery of the enzymes. Then we will study the specific properties of enzymes. We will see how the enzymes work, that is mode of action of enzymes. Then after that we will study different factors which affect the enzyme activity. And finally, we will study about nomenclature and classifications of enzymes. In today's lecture, I will take up first three topics, that is discovery, properties and mode of action. So let's begin with the first topic, that is discovery. What are enzymes? Enzymes are actually, today we know they are the biocatalyst. They catalyze, they make the reactions happen faster. So, this enzyme, how was it discovered? The first enzyme which was discovered was zymase. Now, many scientists have contributed in the discovery of enzymes, like Louis Pasteur. He called them as ferments. He noticed that when yeast is added, there is a very fast reaction taking place, that is fermentation taking very fast. So he called that there are certain ferments which speed up the rate of reaction. Later on, Buchner, he also observed that this reaction, that fastening of the reaction does not take place only inside a living cell. Even in the cell-free medium, it can take place. But the actual contribution of naming or giving term to these biocatalysts as enzymes goes to Kuhn. He actually gave the term enzyme. Enzyme in Latin language is known as enzyme. Zyme stands for yeast. So there is some catalyst in yeast which speeds up the rate of reaction. So the term is given enzyme. And the first enzyme, because it was discovered in yeast, so the first enzyme discovered was zymase. Okay, so this is a very brief history about enzymes. Today there are so many different enzymes known. Lot of work is already done in enzyme. Let us first see the general properties of enzymes. Enzymes, when we study them, we have to take care of three particular properties. One, their catalytic power. Second, their specificity. And third, their regulation. Before coming to these particular properties, let us first see what are actually enzymes chemically. Enzymes are all proteins, that to tertiary proteins, except one enzyme which is known as ribozyme, which is an RNA. Except this ribozyme, all other enzymes are proteinaceous in nature, and that too, they are tertiary proteins. They are globular proteins. Okay, so enzymes, there are certain terms you must know while studying the enzyme. There are terms like, one term is called holoenzyme. Because enzymes are big molecules. So this holoenzyme may be made up of two parts. One is called apoenzyme. The protein part of any big enzyme, a combined enzyme, is called apoenzyme. This is the proteinaceous part. This is having only protein part. And that too, which protein? Tertiary protein. Alright. Now the second part, which is non-protein part of enzyme, is called as cofactor. The non-protein part is called cofactor. This non-protein part can again be of different types. It may be prosthetic group, or it may be coenzyme, or there may be some metal ions. So what is the difference? When some organic part, non-proteinaceous organic part are the parts of enzyme, then they are called as coenzyme. Whereas when some inorganic part is present along with the apoenzyme, then it may be prosthetic group or metal ion. And what is the difference between prosthetic group and metal ion? Prosthetic group are very closely bounded to the enzyme. Very closely bound. Whereas when it is free, metal ions are free, then they are called metal ions. For example, in case of the enzyme carbonase, carboxylase, iron is very tightly bound. Iron is very tightly bound to the enzyme. So that becomes the prosthetic group. But for any enzyme, if some metal ions like calcium ions, zinc ions, they are required, but they are not bound very tightly, they may be free, then they are called as metal ions. Alright? So, there comes the cofactor. So, cofactors may be prosthetic group, metal ions or coenzymes. Whereas apoenzyme is totally the only protein parts. There are certain enzymes where cofactors are not there. Whereas there are certain enzymes where both cofactor as well as apoenzyme is there. Apoenzyme is a part which must be there. Without this, enzyme will not be in existence. But the working of enzyme, for many enzymes require some non-protein parts, which may be called cofactor. Okay, so this was about the holoenzyme, apoenzyme, cofactor. So certain times you may be asked a question based on these terms. So you must keep it in mind. Okay, now we come to the properties. First property of any enzyme is its catalytic power. What is the meaning of catalytic power? What is a catalyst? Catalyst is any substance which accelerates the rate of reaction without participating in it. So all enzymes are catalytic in power. They will speed up the rate of reaction, but they will not participate. Okay. So that is first property, that is catalytic power. It will enhance the rate of reaction. I told you in previous lecture. For example, carbon dioxide and water, when they react in absence of the enzyme carbonic anhydrase, then the rate of reaction is 200 molecules per hour. The production of carbonic acid is at the rate of 200 molecules per hour. But when an enzyme is participating, when an enzyme is there in the conversion, then the speed becomes around 6 lakh molecules of carbonic acid per second. So you can see how fast they make up the reaction. So first property is catalytic power. Now the second property of every enzyme is its specificity. What do you mean by specificity? Specificity means one particular enzyme is specific for a particular substrate. When we say carbonic anhydrase, it will always produce carbonic acid. It will not act to produce any other compound. So they are very specific. For example, when we say lactase, lactase will always act on lactose, nothing else. Same way if we say sucrase, sucrase will always act on sucrose. Okay, so these enzymes are very, very specific with respect to their substrates. Now, third property is its regulation. All these enzymes, they regulate the rate of reaction. All these rate of reactions, they are reversible depending upon the conditions required. So they regulate the rate of reaction. Whenever the conditions are favorable, they will act. Whenever conditions are not favorable, they will not act. So their action or inaction will be depending upon the conditions given. For example, if you keep the milk outside the refrigerator, it gets spoiled very fast because the enzymes act very fast of the microbes. But when you refrigerate it, it is not spoiled. Why? Because the conditions are not favorable for the enzymes of the microorganisms. So they cannot spoil them. So activity will not take place. As a result, the enzyme activity will go down. Okay. Now let us see how they act. What is the mode of action of the enzymes? How enzymes help in accelerating the rate of reaction. Let's see that. Let's now study how the enzymes perform the catalytic action. For any reaction to take place, for example, there are one substrate or many substrates. So the substrates are there. These substrates have to undergo the transition state before becoming the product. So that energy, that transition energy is called as the activation energy. So if you see, the ground state of substrate is very low. Low potential energy is there. And the ground state potential energy of product is even lower. But for conversion from substrate to product in any chemical reaction, a lot of potential energy is required. And that potential energy is actually the activation energy or the transition energy, which is very high. So if you see the condition, there are two conditions. This condition when enzyme is not there, and this second condition when enzyme is there. You can see where black curve is shown and blue curve is shown. When there is a black curve, you can see the transition energy required for developing a substrate into product is very high. You can say very high activation energy is required. So very few molecules, you can see only 3 molecules can participate in that reaction, because that much free energy is not available for all the molecules. Very few molecules have that energy. So the rate of reaction is very slow because very few molecules are participating in the reaction. But what is this enzyme doing? Enzyme is lowering the activation energy. So for example, if you have to take out something from very high uh, um, very steep height. So what will you do? You have to take a lot of energy, and you have to apply a lot of energy. You have to jump very high, and then you get that product down. But if you get something to stand on, and then you can very, with less amount of energy only, you can bring that substance down. The same thing is done by enzyme. Enzyme is actually lowering the activation energy to reach the transition state. So even at lower energy, the transition state is achieved. So here you can see so many molecules. These molecules which were earlier were not able to participate in the reaction. They were not converting into product. Now these all molecules can also participate. Because so many molecules are participating, so lot of fast reactions take place, and the rate of reaction increases. And this is how the process takes place. This is actually lowering up of the activation energy. This diagram, this graph is very important. Please practice it. How enzymes speed up the rate of reaction. Okay, so this activation energy graph is very important from exam point of view. Now let us study what are the different modes and mechanisms of enzyme actions. See, enzymes are nothing but tertiary proteins. They have certain pockets, which these pockets are called as active sites. These are active sites. Suppose these two are the active sites. These active sites are specific to their substrate. So two mechanisms are very common. First mechanism is called lock and key hypothesis. According to this was given by Arrhenius. He explained the lock and key hypothesis. As we know, for every lock there is a specific key. The hole in the lock is complementary to the key. Same way, substrate and enzymes are complementary. These active sites are complementary to a specific substrate. Okay, so when enzyme and substrate come in contact, by lowering the activation energy, a transition complex is produced, which is called as substrate enzyme complex, which is temporary state. And this enzyme, by lowering the activation energy, it breaks down the substrate and converts into product. But as I told you, enzymes don't participate in the process. They only enhance the rate of reaction. So as a result, this enzyme can be used again for next reaction and the product is produced. This is the most common hypothesis, which is called as lock and key mechanism. Okay. But nowadays, seeing a very important property of enzymes, which is called as that is competitive inhibitions. There are certain inhibitors which can affect the rate of reaction. So these inhibitors compete for the active sites. It has been observed that these active sites are not very fixed as mentioned by lock and key hypothesis. So this hypothesis has now been modified and it is called induced fit theory, which was given by Coastland. According to him, the active sites are present in the enzymes, but they are not very rigid. In fact, they are flexible. And due to their flexibility, they can many a times act on more than one type of substrate. For example, if you see this is substrate 1, this is substrate 2, having different specific sites where the active sites will bind. But this enzyme, depending upon the substrate, it can modify its active sites and can act on both the substrates and can produce the products. This is called an induced fit hypothesis. Okay. So today we have studied about some properties of enzymes, and in next lecture we will study more about these properties. We will study what are the factors which are affecting the rate of reactions in enzymes, and we will also study the classification of enzymes. Okay, how the enzymes are named and how the enzymes are classified. Okay, thank you very much.